Good evening, everyone. We're going to get started with the drugs portion of the evening. To all of you who are joining virtually, thank you. Good morning, good afternoon, good evening to you as well. So my name is Zad Tanvir, and I'm the Secretariat for the Working Group on New Drugs. I'm very happy to be here with you in Copenhagen. I have to say, it's been a bumpy year, so I wasn't sure if this meeting was going to happen, but I'm very happy that we're all together and that our virtual participants can enjoy it as well. I'm going to introduce one of our co-chairs for the Working Group on New Drugs, Melvin Spiegelman, and he'll be addressing all of you to give you some welcoming remarks. You have to press that button.
It's on, I think. The stalwarts. How many of you have been here all day? Okay, that's great. No, thank you all for coming. I'm sure you've heard, and I apologize, I wasn't able to be here. I just got off the plane not that long ago. But it's really great to see everybody's still here, or people still here, in spite of, I'm sure you heard this morning in the introductions, for those of you who are here, that these are, somewhat challenging times for the working groups, as well as for so many other parts of the ecosystem in TB R&D. But we will prevail. I have no doubt about that, and things will improve, I believe, significantly. It'll take some time, but we will get there, given the really commitment of everybody. First, let me just bring greetings and regrets also from Barbara Wong, the co-chair, who really does all chairs' work. I don't do anything, Barbara does all the work, just to, put it out there. And unfortunately, Barbara was not able to join us today, but she certainly is here in spirit, and she may be online. I don't know. Yeah. So hi, Barbara, and thank you.
Yes, hi, I'm here.
Great.
Hello, everyone.
So also, I just want to set a tone a little bit for today, because what we'll all be hearing. It really is a tragedy in a sense to be hitting the semi-brick wall of funding at a time when the pipeline, certainly for drugs, has never looked, in my opinion, rosier than what it looks like today. And that's part of really the reason for optimism, because I think this is being further and further recognized by so many other partners around the world, who really see the value of what all of you all and all the presenters here are working on, and what's going to be done over the next ensuing few years. So I think we all should really take pride in that, because clearly it's been an effort that transcends countries, institutions, people, etc. So with that, I'm going to turn it over back to Zad to introduce the speakers, and really I think it's going to be an exciting next session. Thank you.
Thanks so much, Mel. Can everyone hear me okay? Yeah. One request we had from Barbara Lon, who is here virtually, was to just show you this global TB drug pipeline, who, Barbara really is the brains behind putting this together, and with the help of all of the WGD membership, we're able to track what's going on globally in TB drugs. So this is our PowerPoint slide that shows all of the new chemical entities. And I very much like to emphasize that it is a living document. If you know of anything that's not on here, or advances that should be on here, please let us know. It's through doing our own research, attending these meetings, talking to people, that we're able to populate this slide. So we just want to, let you know you can always come up to us and with any edits or feedback. So our first speaker today is Nyasha Bakare from Johnson & Johnson. And I'm just gonna hand over the mic to her and we can begin.
Thanks, Said. Hi, good evening, everyone. Nice to be here today. Thank you for the invitation. I'm happy to present on behalf of our team at J&J some of the current work that we're doing on bedaquiline long-acting injectable clinical development program. So interest in long-acting formulations has been increasing in recent times, not only in the HIV domain for both treatment and prevention, but also with regards to the potential that a long-acting formulation could have for TB control. There are different possible uses for a long-acting formulation in the setting of TB. Those could include TPT, so preventive treatment for latent TB infection, or to incorporate a long-acting injectable into a treatment regimen for TB disease. With regards to what that could possibly look like in a preventive setting, it could be possible to have a once-off shot that could cure latent infection. In the setting of treatment, you could arrive at an ultra-short regimen by incorporating a long-acting injectable into a regimen, perhaps into the continuation phase to shorten treatment. Now, of course, in the treatment setting in particular, it would be important to have companion drugs together, because you wouldn't want to have a single drug in that continuation phase. But in the setting of prevention, it could be interesting indeed to have a single drug that could be used to achieve cure. There's great potential, of course, to improve adherence if you were to use only a one-time required treatment, as in the setting of prevention. But this, of course, has to be balanced with the potential acceptability of intramuscular injections, of which, of course, the TB field is very familiar. And there would need to be, for any future research, a lot of attention paid to looking at the acceptability of injections in various populations, of course, particularly in younger children, and looking in particular at patient preference studies and incorporating patient-reported outcomes into ongoing research. So the top right of the slide shows an excerpt from a review from 2019 on the history of preventive treatment in TB. And what you can see is that the duration of treatment, of course, has really been shortened over the years to the currently recommended treatments now by WHO being 3 HP, so 3 months of isoniazid and rifapentine weekly, or even 1 HP, which is a month of isoniazid and rifapentine daily. And so there have been great advances here in making these treatments easier to administer for prevention. When it comes to bedaquiline, bedaquiline actually has some CMC properties that make it a good candidate for developing a long-acting formulation. It has a low aqueous solubility, it has a low plasma clearance, and it's efficacious at lower exposures. So it's possible that a long-acting bedaquiline formulation could also be used against both drug-susceptible and drug-resistant isolates, and so you could have a pan-TPT treatment that could be used potentially as monotherapy. This would ensure higher completion rates for prevention and, as mentioned, could shorten TPT to a single visit. It's also helpful, of course, that we now have a lot of data on the side effect profile of bedaquiline, which is expected to be quite favorable also in this setting, particularly as one would anticipate that lower exposures are needed for preventive settings than for treatment. And the bedaquiline long-acting formulation specifically has been formulated to not require cold chain for storage, which is another advantage. So a lot of work has been done to develop translational PK/PD models of both oral and long-acting bedaquiline formulations. And these have been applied and validated preclinical models in mice for latent infection. And these were the basis for deriving a target exposure to use a long-acting bedaquiline formulation in a first-in-human study. And the target exposure there was specifically trying to arrive at the same CFU decline that you would expect after 3 months of 3-HP. So this slide shows you the current design of our ongoing first-in-human study. It's a single ascending dose study with healthy participants enrolling at a single site in Austria. The participants will be enrolled into 3 groups, the low-dose group, a mid-dose group, and a high-dose group. Each of them have a sentinel cohort initially for safety, and then the rest of the participants are enrolled if the safety profile is looking favorable. So there are 3 dose levels, as I mentioned, by volume. The lowest dose is 2.5 ml, the mid dose is 5 ml, and the highest dose is 2.5 ml. And these are intramuscular injections administered into the ventrogluteal muscle. The primary endpoints for this study include the various PK parameters. The participants are followed for 52 weeks after the injection. And so various PK parameters are evaluated to determine exposure over that period of time. And of course, there's close follow-up of safety and paying particular attention in this context to local injection site reactions and adverse events of special interest. The full results of the interim analysis of this study, which are now available, will be presented on Wednesday by my colleague Vivian Cox in the Union CDC late breaker session. I hope many of you will be able to join. And with regards to the further completion of the study, the last patient out should be in May next year, so that we have a final analysis available next year as well. Just some further details on the actual follow-up during the study. So from a safety standpoint, we're certainly looking very closely, looking at all the usual SAEs and grade 3 or higher adverse events, paying attention to the adverse events of special interest that are specific to bedaquiline based on the known safety profile of QT prolongation and transaminase elevations, but also looking at hypersensitivity reactions and solicited injection site reactions to make sure that we understand the safety profile in this context. The participants are also asked to complete VAS or visual analog scales for pain to rate the level of pain as shown on the bottom left on the scale from 0 to 100 immediately after the injection and then also subsequently. And finally, the study includes a participant questionnaire, which is a kind of preference questionnaire asking the participants whether they would prefer a treatment that consists of pills taken by mouth, that is looking either like a 1 HP or a 3 HP type of treatment versus the administration of 2 injections given intramuscularly as in this context. So those data will also be collected in the context of this study. So I will close just by pointing you to the late breaker session as mentioned on Wednesday, where you can hear the full results of the study. And thank you very much for your attention.
So we do have some time for questions, if anyone in the room would like to ask one.
Thank you very much, very interesting. If you could go back 1 slide, in this questionnaire where you mentioned you asked about which treatment they would choose, could you, maybe I missed it. So do you specify duration? So do they need to choose between, okay, this treatment by mouth without pain for how long versus this injection that may be a bit painful for?
Yeah, so this questionnaire is administered at different time points, either before they received any injection or then also after they started to receive injections in the course of the study, asking them if they would prefer to receive as a treatment, hypothetically, injections of this kind as they're receiving in the context of the study, but actually two injections of 5 ml versus taking tablets for the specified amount of time as you would in a 3 HP or 1 HP regimen. So that's the comparison that they're asked to make.
Another question this year.
Thank you, Yasha here. Do you think that lidocaine HVCC in each syringe would be compatible like it is for amikacin? Seriously reduces the injection-associated pain and there's no pharmacological interaction.
It's certainly something that could be considered. I think let's wait and see what the readout is for the local tolerability for this particular formulation. I think it also depends on what the formulation looks like, right? This is an aqueous formulation, so I think we'll wait to see indeed what the data look like, and then those are possible mitigations indeed. Yeah.
One more question. Okay. I'm gonna get all my steps in.
It is very interesting. Is there any evaluation of immunologic factors regarding disease TBD, and also how the result regarding the outcome of TB relapse or TB become develop active TB?
Thank you for the question. The participants included in the study are all healthy participants, so it had to be excluded that they had any TB infection in order to be able to enroll into the study. So this is just a first in human study in volunteers. So therefore, no further follow-up of immunological parameters.
Thank you so much. Thank you to Nyasha. Next, we'll be hearing from the Gates Medical Research Institute from Chris Vinard.
Good evening. Thanks very much. Thanks for sticking around, and I'm really pleased to share the updates of what we're doing with the TB drug portfolio at the Gates MRI. So I'll go over sort of two sections. So first will be a bit on our phase 2b/2c trial, pan-TB trial in terms of regimen development, what our TRP is aiming for, and again, an overview of our pan-TB collaboration and trial design. And touching on the pan-TB trial itself, which is also known as TBD-06-201. And then I'll get into some individual updates of TB drugs candidates in our portfolio. And that includes TBD-09, also known as MK7762, TBD-10, MK3854, TBD-11, known as CLB-073, and nicotinamide. So this is what we are aiming for in terms of our pan-TB collaboration target regimen profile. Emphasizing test and treat, not having a need for upfront BST for initiation of treatment. Shorter, 3 months or less. An acceptable safety profile without requirements for baseline or ongoing safety monitoring. Simpler, efficacious in terms of being non-inferior to standard of care, and affordable. So this is our North Star in terms of our TRP that we aim for. So the pan-TB trial, TBD-06-201, is a trial to evaluate two regimens, delamanid, bedaquiline, carbidopa, cetazolid, and protomanid, bedaquiline, carbidopa, cetazolid, in comparison to standard of care, HRZE. This was a two-stage phase 2bc trial to look for a regimen of a 3-month or less treatment duration. It was 2 stages, and stage 1 included arms of 4 months in length of DBQS or PBQS versus HRZE. And so the innovations in this design was stage de-risking for treatment shortening, looking at phase 3 endpoints early, which were end of treatment 12-month unfavorable outcomes, the use of whole genome sequencing, novel biomarkers and evaluation, person-centered treatment support, and a stop treatment and watch framework. So these were all innovations introduced into the trial with the stage 1 of the trial. The updates, the trial was launched in South Africa, July 2023, and I have the QR code here that can link to a CT.gov posting. 11 sites eventually participated in South Africa and the Philippines. And I'm not saying a lot here because there is an entire symposium coming up on Wednesday devoted to the PAN-TB partnership and this trial. And so the symposium Progress Towards the PAN-TB Regimen, Insights from this trial. So I encourage you all to attend if you're able to. And the speakers, just to give a bit of an advertisement for what the speakers will be talking about during that trial session. Shifting gears to our individual drugs in the drug development, clinical development pipeline. First is TBD09. It's an oxazolidinone. It was discovered by Merck with support from the NIAID and the TB Drug Accelerator. So the phase 1, first in human, SAD/MAD food effect has been completed. That trial is the name TBD09-101, where we give lots of letters and numbers to our trials. The CT.gov posting is at the top, and this will also be presented on Wednesday at a late breaker abstract, and I have the QR code for the session, so I encourage you to come. The full set of data reported from that phase 1 trial. We have an additional phase 1 trial that is currently ongoing with TBD09, and that's a trial with up to 84 days, 12 weeks of daily dosing, evaluating 2 dose levels in healthy participants. So that trial is fully enrolled and follow-up is underway as we speak. And that CT.gov posting is there for you as well. And we are now preparing for our phase 2 trial of TBD09 to look at safety, PK, and bactericidal activity in participants with tuberculosis. TBD10, also a protein synthesis inhibitor, but structurally diverse from an oxazolidinone, also discovered by Merck with an IND through the TB Drug Accelerator. For TBD10, the IND enabling studies have been completed, but this is a backup compound to TBD09. So we are waiting to see how TBD09 continues to perform before making decisions on TBD10. TBD11, this is an agonist of MTB adenylyl cyclase, RV1625C. Essentially, it inhibits MTB's ability to metabolize cholesterol. It was discovered by Caliber with Cornell through the TB Drug Accelerator. And this is another phase 1 trial that is currently underway. It includes single ascending dose, cohorts food effect, multiple ascending dose, and a crossover to evaluate a tablet formulation of TBD11. So another trial currently underway in phase 1. And then finally, in terms of clinical trial activity moving forward, nicotinamide. So nicotinamide is a water-soluble amide of vitamin B3. It was actually evaluated very early on as a potential TB therapeutic. And it had a problem at the time, which was antagonism with isoniazid. Couldn't really put them together. It was discovered pretty early. And so nicotinamide was really put aside. We've seen some very interesting non-clinical data demonstrating that nicotinamide can be added to a backbone that includes BPA, bedaquiline protomidate, or bedaquiline protomidate sutazolid, and contributes in the Kramnik mouse model, which is the data shown here, contributes to sterilizing activity when added to those background regimens with those important mechanisms of action. And we also have non-clinical data demonstrating that this contribution is preserved when the NTB strain is resistant to pyrazinamide, which is quite important. So we are also preparing a phase 2 trial of nicotinamide, and those activities are currently underway to evaluate nicotinamide's contribution to treatment of TB patients. That brings me to the end of the updates. And so I just to acknowledge PAN-TB partners, IMI, Merck, Caliber, NIH, the Foundation for Neglected Disease Research, and the TB Drug Accelerator, and happy to take questions. Thank you.
Yeah. If you'd like to.
Yeah, I'll repeat. That's fine.
Yeah. Very interesting. Do you have any data on the nicotinamide to undernourished, well-nourished mice, or these recent experiments on nicotinamide?
Yeah, so the question is on the nicotinamide data on the basic nutritional status of the mouse, and if that underlying nutritional status influences the data. I think, so I'm not ready to present any of that data here in the session. We are generating data like that in terms of looking at the role of nicotinamide under different scenarios, but that's not something, it's an important question, obviously, because of the role of nicotinamide in malnourishment.
Any other questions? Yeah, if you're welcome to.
What happens with the ozonic acid impact? Like, does that just kind of get dissipated because you're doing a combination of drugs, or did you reformulate the nicotinamide so it didn't impact that pathway?
So the question is, why do we see the activity of nicotinamide in the mouse model, if I'm understanding correctly, where the MTB strain is resistant to oh, isoniazid, yes. Excellent. Thank you for that. It's an important question, and thank you. So part of the interest in the potential utility of nicotinamide is recognizing that isoniazid may not need to be a part of a next generation first-line pan-TB regimen. So if we can sidestep the isoniazid nicotinamide challenge, then we've got a potential therapeutic agent with nicotinamide with a different background that may include dialquinolines, nitromidazoles, et cetera. Yeah, thank you. It's an important question. Thank you.
We have time for one more. All the questions come from the top, I see. Sure. Well, I'm here now.
Hi, thanks, Chris. Could you, I'm sorry if I missed it, could you comment on the trial design for the phase 2 for TBD09 and what you're trying to demonstrate there?
Yeah, it's a, thank you. So the trial design for TBD09 phase 2 is a dose ranging trial. We think the way to establish dose response, exposure response with TBD09 is actually with a background combination. So we're not doing a monotherapy 2-week EBA type of trial design. We're treating for 28 days, but with a background regimen that TBD09 has added across a range of doses to establish dose response that we think we can move forward with then in future studies. Thank you.
Well, thank you so much, Chris. That's very exciting work. Our next speaker is coming from Unite for TB, Jody Schildkraut. Jody, welcome.
Hi everyone, thanks for having us. I'm very happy to be updating on behalf of the Unite for TB consortium. So just quickly, a couple of minutes on, seconds, sorry, on what Unite for TB is again. So Unite for TB is an innovative multidisciplinary phase 2 consortium with the goal of setting a new standard for anti-TB regimen development. It started in 2021 and it's set to run 2028. We have a core team of leadership, so coordinated by Derek Sloan, who I work with in Radboud in the Netherlands, and then scientific leadership from Michael Hilscher, project leadership from David Barros at GSK, and we have rotating clinical leadership from Guy Thwaites, Nick Payson, and Christoph Wagner. So who are Unite for TB? So Unite for TB, as I said, is a consortium. It's made up of 30 partners from 13 countries. 25 of which are public partners, and the other 5 are what we call FPO-associated partners. So this is a public-private partnership funded by the Innovative Health Initiative by the European Commission. And we're also very happy to have a great set of clinical trial sites. So over 30 sites in 13 countries on 4 continents. And so you can see the flags of the countries that are represented here. And so to zoom in a little bit on the clinical trials that are ongoing, so at the moment we have 4 protocols running, or set to run under Unite for TB, and we have a 5th one in development that we hope to be able to update on soon. So we have 1 in the kind of early phase 2A phase, which is the trial looking at alpha-vector epionamide, and then 3 in the later phase 2B/C stage, 1 looking at BT7043, and 2 multi-arm, multi-stage platform designs looking at multiple novel chemical entities that you can see listed here. So I will run through them all one by one, but unfortunately, because of the time, it will just be a very high level overview. So first, Enable. So Enable is a clinical trial looking at ethionamide alpha-bacteria. So alpha-bacteria is actually an ethionamide booster. So that goal is to be able to dose ethionamide lower by also dosing with alpha-bacteria. So this trial is really looking at what the optimal combination of alpha-bacteria and ethionamide is. It's a two-stage trial. So in cohort 1, we look at different combinations of alpha-bacteria and ethionamide. So ethionamide either at 125 milligrams, 250 milligrams. And then cohort 2, adding to the alpha-bacter ethionamide, rifampicin, pyrazinamide, and ethambutol, which is to see how this drug is in combination, and both of those compared to the standard regimen. It's a 14-day trial, so it's an early bactericidal trial looking at CFUs and TTP. And recruitment completed this. It recruited in South Africa, so it tasked UCTLI in Cape Town. And we're hoping to have the first data from this trial maybe by Q2 of next year. The second trial is the Decision trial. So this is a dose optimization trial for BTZ-043. So it's a trial looking at 3 different doses of BTZ, so either 500 milligrams, 1,000 milligrams, or 1,500 milligrams once daily, in combination with benacolin and delamanid. And looking at the efficacy, safety, tolerability, PK, and the dose exposure response of these drugs. And the primary efficacy endpoint was a microarterial load over time. So the results of this are actually presented this Friday. So if you're interested, please go. It was presented in room B4B by my colleague Michael Hosha. So very excited to have the first results coming out of the United Kingdom trials. The third trial is the Paradigm 2B trial. So this is what we call our flagship trial. So this is a large multi-arm, multi-stage trial. It's a seamless phase 2BC trial as well. So in phase 2B, we're looking at different regimens, and phase 2C, we'll be looking at different durations. So for sake of time, I'm not going to run through all the different arms that we have, but phase 2B wave 1 is what we call, which you can see here are the most left regimens, has actually finished recruitments already, and those were all combinations of with BTZ-043, organfebrol as new chemical entities, and also one arm in which they were combined with each other. So we're currently waiting for the data to come in to be able to do the interim analysis. After that, we need the week 16 TTP slope as an efficacy outcome. And we'll take that together with safety and hopefully other endpoints like PK and some biomarker work, and rank which regimen is looking most promising to continue into duration selection. And then the protocol now stipulates durations from 8 weeks up until 16 weeks with 2-week increments. So while we are waiting for that data to come in to make the decision for duration selection, we have initiated a second wave of regimen evaluation. And you can see here where the arrow is, that's where we are right now. And then we're bringing in some more new drugs. So looking at carbidepistats and also at, again, at BTZ and gamcaparol. So just a little update on where we are with the Paradigm 2B trial. So as I said, we've completed wave 1 recruitment, and you can see on the left is the recruitment for wave 2. So that is set to finish recruitment early next year. So by March of next year, we'll finish that, all of the regimens that we will be trialing, and that's when we hope to start with our duration ranging. And on the right, you can see all the trial sites that are coming into United Kingdom are already in. So in green are all the sites that are already active. In light green are the sites that will become active in the next month. So we'll continue contribute to regimen selection. And then orange, we have all the sites that we hope to bring in as well in the phase 2C for duration ranging. And yeah, as you can see here, some key timelines, but just to say the database lock for final analysis of all of the trial results is set for Q2 of 2028. And then just one final trial slide. So this is the STEP2C trial. This is actually a trial that's currently running under the Panacea Consortium. And stages 1 and 2 are still ongoing. So that have recruitment completed, but are still in follow-up. So stage 1 was looking at optimized doses of rifampicin and optimized doses of pyrazinamide. And in stage 2, there was also an arm in which BTZ was being introduced as an alternative for ethambutol with a microbiological endpoint. So as I said, that was running under Panacea, but we're very happy to have been able to adopt this trial into the United Kingdom Consortium, so to continue the great work it's doing. And we have a stage 3 protocol is being designed, and there we will start looking at new regimens, so benacolin sparing regimens. So that will be a regimen of protominid, ganfebrol, BTZ and delpazolid given for 6 months, and also continuing the work from the Enable trial, looking at a regimen with alpha-vector ethionomide and isoniazid monoresistant regimen. So that will be the LE with RZE for 8 weeks and then 18 weeks of rifampicin and isoniazid. This trial is set to start in early 2026, so in January, which we're very excited about. So yeah, this is stage 3. So just a summary, so the preparatory work during the first phase of Unite has progressed to trial delivery. As you can see, we have 4 trials with protocols already set, many of them running, and a 5th one under development. Decision will be presented this Friday. Enable has fully recruited with results expected soon. Recruitment in Paradigm to be advanced and performed beyond the first wave of regimen selection, with step 2C set to initiate in early 2026. And within Unite, therefore, we're aiming to undertake these phase 2 evaluation of at least 5 new chemical entities in these kind of novel study designs to integrate adaptive trial design, biomarkers, and analysis. Thank you.
Thank you, Jody. Does anyone have any questions? If not, we can move on. Thank you so much. So our next speaker, thank you. Our next speaker, unfortunately, his plane was delayed. So I actually gave him 2 options. One is the Zoom. So Nick, are you on the Zoom with us? I can't actually see who's on. If not, Eric, do you think you can take over for Nick? This is Eric Nuremberger, who's going to be one of our speakers later on in the agenda. He has graciously agreed to take over Nick's slides. These are regarding the TB-TC Study 38 CRUSH TB trial. Thanks, Eric.
Sure. Okay, thanks, and bear with me. This is a new assignment for me. Try and do this quickly. So I'm going to talk about the CRUSH TB trial. As you know, this is a TBTC sponsored trial. The TBCC is charged with doing programmatically relevant research, most relevant to the United States. We are working in 21 sites in 8 different countries. However, for the CRUSH TB trial in particular, there are 6 sites enrolling into wave 1, and they're shown here in blue. This is a phase 2C randomized open label adaptive trial that's looking at 2 regimens plus the control regimen. The 2 investigational regimens are based on a bedaquiline, moxifloxacin, and pyrazinamide backbone. And will be 4 months in duration compared to the 6-month standard of care. Here is the schematic. You can see those ending after 17 weeks of treatment compared to the standard control. So this is what we're referring to as wave 1. They'll be followed up for relapse with a maximum follow-up period of 78 weeks. However, while waiting for the analyses of those results, we're going ahead and enrolling into wave 1A, which is to extend enrollment into certain priority populations that don't normally get represented in large numbers at trials in these sites. And I'll show you who that includes in a moment. And then starting to think about what wave 2 should look like, what regimens will be worth looking at perhaps in wave 2. Happy to announce that wave 1 is fully enrolled. 292 participants enrolled as of about a month or month and a half ago. We have passed the first interim PK and safety analysis, mainly looking at bedaquiline PK and safety, and we're advised to proceed. The next DSMB meeting is to happen sometime soon. We'll look at the interim efficacy, and hopefully by this time a year from now, we'll be presenting at a minimum the top line results and hopefully the final phase 2C outcomes. So I think you may have seen some of this last year. The consortium has been working together with the Predictor Consortium to help develop some expectations about what the relative efficacy of these regimens might look like. This is modeling that's been done by Rada Savic and her team, looking at the BMZ-RB, which is BMZ plus rifabutin, and the BMZ plus delamitid investigational regimens compared to HPZM. These are time to relapse prevention in the mouse model, VALP-C relapsing mouse model, and you can appreciate that both BMZ-RB and BMZ-D are expected to have a shorter treatment duration needed to prevent relapse. And then similar site of action predictions for drug exposure, both the experimental regimens on the bottom expected to have effective drug concentrations in caesium for at least 2 drugs, being bedaquiline and moxifloxacin for the majority of the dosing interval, and rifabutin as well in that rifabutin arm. So expected superior activity at the site of action in caseous lesions. So wave 1A is again this extended enrollment of priority populations. Here you can see in the table, those are individuals under age 18 or at or above age 50, participants with diabetes and participants with HIV. Fairly modest numbers enrolled during wave 1 will be expanded during wave 1A, which is starting in January. And now for wave 2, we've started to think about what sort of novel regimens we would love to try and employ. And so there have been a number of considerations here. One, simply taking the best looking arm or arms from wave 1 and evaluating that as a 3-month regimen as opposed to a 4-month regimen. Adding arms with lower pyrazinamide doses to try and improve tolerability, or perhaps some other regimens that might be PZA sparing. And then probably a little longer down the pipe, looking at potentially long-acting injectables or other drugs from other partners. So we're going to present a little bit of the preclinical evidence that the Predictor Consortium has been generating for some of these pyrazinamide-containing or pyrazinamide-sparing regimens. The S here in these animal studies is sorfequilin, or TBHA 876. And so we've looked at regimens that would essentially swap bedaquiline out for TBHA 876. Given that drug has been more potent in animal models, has good distribution into caesium, where it's again more potent and distributes this more quickly perhaps than bedaquiline into rabbit lesions as well. And then several pyrazinamide sparing regimens based on an SPAM backbone, looking at rifabutin, sutazolid, or cabodebastat. So questions like, if we just substitute 876, does that look better than bedaquiline? Is sutazolid a better 4th drug in the SMZ backbone? Could a lower PZA dose preserve efficacy while being more tolerable? And could protominid substitute for pyrazinamide in a pyrazinamide sparing regimen? We've done some actual studies as well as some predictions. These relapse predictions are based on actual mouse model data, suggesting that both that actually the best regimens were the enhanced regimens with sorfequilin combined with the rifabutin or delamitid based regimens that are looking at in wave 1. Their substitution of sutazolid was not quite as sterilizing in these studies. And the PZA dose reduction in these 2 arms being compared here really didn't seem to have any measurable effect on sterilizing activity, suggesting lower, perhaps better synergy and lower pyrazinamide exposures could be afforded. The SPAM-based regimens did not perform as well as the pyrazinamide SMZ-based regimens, but relative to the BPA-MZ reference, performed about as well in the case of the carbodepistat and sutazolid containing combos. We have looked at Kramnik mice as well and see faster reduction in CFU counts and earlier reduction in relapse proportions, again with the SMZRB regimen compared to BMZRB, although ultimately, perhaps in the hardest to sterilize animals, there was not a clear signal for benefit. But we are again seeing superior sterilizing effects of rifabutin over sutazolid and pyrazinamide over protominid in these combos. So in summary, wave one enrollment is complete. The top line wave one results are expected about a year from now, hopefully less. We're enrolling into wave 1A, expanding into priority populations, and looking forward to wave two. Thank you.
Thank you so much, Eric, especially running up here and doing that last minute. Does anyone have any? I'm not sure how much we can answer, but if anyone has any questions, we can try. If not, I know we are moving a little bit behind, so we'll just move on to the next speaker. Thank you, Eric, and we'll see him again a little later down the line. So our next speaker is from Otsuka Pharmaceutical, Titus Davala. Titus, welcome. Yes.
Thank you very much for having me. Yeah, my name is Titus Davala, and I'm the newest member of the Global Clinical Development Team at Otsuka. And I'm very excited to be the one to present to you an update on where we are with the clinical development for carbodepistat. I just wanted to start by saying that it is still a compound that is still undergoing investigations and not yet registered. So what is carbodepistat? You have seen it in a number of presentations today, but just to give you a background, it is a DPRE1 inhibitor, a novel mechanism, and our studies so far have demonstrated that it has antimicrobacterial potency, and this potency is cutting across resistance profiles and also has shown potential for treatment shortening. One critical piece of steps that we have taken is the completion of EBA studies, which have been successful and motivated us to progress to further studies. This particular slide is showing data to do with an important question about whether the drug can reach the disease site. So you have these slides at the end of the meeting, but the main message here is that carbodepistat can penetrate even the necrotic calcium, which is the most difficult in terms of TB pathology, and maintains sufficient concentration required to address treatment shortening as well as TB pathology in general. This evidence together motivated us to progress to phase 2B/C trials. So the phase 2B/C is what's shown on the slide. We had 4 treatment arms. The first 3 treatment arms that you can see there are containing carbodepistat, delamanid, and bedaquiline with a 4-month treatment duration. The doses are 10 milligrams, 30 milligrams, and 90 milligrams. The control arm is RHZ, which is at a standard duration of 6 months. The outcomes, the primary outcomes are safety using standard definitions and proportion achieving sputum cardiac conversion at the end of treatment. Looking ahead, we wanted to still explore long-term outcomes. So we had an exploratory endpoint looking at that. So what did we find in terms of safety? With respect to safety, there were no serious adverse events. The mild and moderate adverse events that we identified did not have any dose response. We looked specifically at cardiac toxicity, and there was no warning signal. And also looking at liver toxicity, as you can see from the two graphs that are there was no concern as well through to the end of treatment. How about efficacy? So with respect to efficacy, the headline result is that there was non-inferiority achieved the 4-month DBQ regimen versus the 6-month RHEZ regimen, as shown in the first line of that slide. With respect to long-term efficacy, which was an exploratory endpoint to guide us in terms of how we move forward, phase 3, there were numerically more relapse cases in the DBQ arms compared to the RHEZ. Putting all these results together, we interpreted that the DBQ backbone is strong enough to provide bacterial, to reduce bacterial burden. But in terms of durability of cure, we needed to optimize the regimen. So how are we proceeding to phase 3, which has just started recruiting at the moment? We have added an additional compound, which is MOXIE, for patients with fluoroquinone-sensitive disease. And for patients with fluoroquinone-resistant disease, we have extended the duration from the 4 months we were using to 6 months. This brings me now to the phase 3 trial, which we're calling Quantum, and has just started recruiting participants. We are recruiting patients who are rifampicin resistant, who have rifampicin resistant TB, and in keeping with the current reality of managing drug-resistant TB, we are recruiting using 2 cohorts. One cohort focused on patients with fluoroquinone-susceptible TB, and you can see the treatment arms that we are randomizing to. The one question which you may be asking is why we're using pretormanide and not delamanide, which we used in phase 2. We're using pretormanide here because we want to mirror, as you can see, the standard of care. The fluoroquinone-resistant group is receiving the treatment as described earlier, BPA-Q, but for a longer duration, 6 months. The efficacy outcome is unfavorable outcome using the standard definition, and also we're measuring safety and tolerability using treatment adverse events. We're going beyond the traditional TB clinical endpoints to look at patient experience. Using these tools, we are hoping to understand patient experiences. This is very important to understand from a patient perspective, because a shorter treatment is only better if it doesn't cause worse side effects. In terms of inclusion criteria, the key factor that I wanted to highlight here is that we are starting with including adolescents, which brings us towards pediatric development. And we're also including participants living with HIV using the criteria described here, which is more relaxed than what we did using in phase 2. We are setting up TNGS at country level to facilitate exclusion of patients with benaglin resistance and other drugs. I wanted to close by saying that we are working on the pediatric development plan. Traditionally in TB, children have had to wait 5 to 10 years from the time that adult development has completed. We are breaking this tradition with cabotegravir. Our integrated development strategy starts with inclusion of adolescents in the phase 3 trial through to completing the steps shown on the slide to conducting a clinical study within younger children, with the time difference between adult and pediatric development completion being reduced significantly. Thank you so much, and I can take questions.
Do we have any questions? Or if you wanted to speak it, we'll repeat it.
I'm David Lambert, University. Very interesting talk. Thank you. So I'm interested in where your relapses were in your trial, where you enrolled them. We published in 2013 in nutrigenetics that mutations in the TPA pathway were seen cause either low or high level methanometabolic resistance, depending on whether mutations are present, and that in sub-Saharan African countries, there were more often mutations in 3806C, RB3806C, which is in the TPA pathway. I'm wondering if your relapses were potentially in people that had mutations, whose back TB had mutations in that pathway, 3806C.
I don't have specific details in terms of mutations, but in terms of geographical location, this study was done in sub-Saharan Africa. So
Worth looking.
it will be interesting to see your data. Yeah, thank you.
Well, thank you. It's great that you mentioned the pediatric so early. It's not going to be global alone. It's in present. So, how about the other compounds?
So the question is about the pediatric program and if it's going to be in combination with other drugs in the regimen.
We are still working out a specific pathway, but yes, that's the key is to understand which drugs are currently being used in pediatrics in routine care, and then how best you can bring in the new carbodepistat to strengthen those existing regimens. But this is something that's still under a lot of discussion within the organization. And yeah.
We have time for one more question. Yeah, please. Sure. Yes.
Yeah, thank you. How do you want to balance between, you said, a longer duration because of to reduce the side effects of the shorter duration drugs, but again, there's the issue of adherence. How would you balance that in the extended longer duration?
So the question is about balancing between side effects and duration of treatment. This is a very important question, and it does really come straight to why we are doing the patient-reported outcomes and putting a lot of emphasis, investing in a whole 4 outcome measures, just focusing on that. We hope that we'll have expertise to look at that kind of data, which can tell us in the end how patients themselves differentiate and how patients tease out the importance of the regimen. And hopefully that's going to be useful also for a program as well as WHO. Yeah.
Thank you so much, Titus. Appreciate it. Our next speaker will be Natalia Servina from TB Alliance. She'll be giving an update on the TB Alliance pipeline.
Okay. Well, thank you. It's really very exciting that this actually has been managed and that this is happening today. I think it's a credit to organizers. So I will give an update on TB several aspects of new developments in TB Alliance pipeline. Specifically, I would like to talk about our dark vanillins update on TBJ 587 and TBJ 876, as well as an update on Tilacibek Q203, which is a newer addition to our clinical pipeline. The dark vanillins, we have 2 dark vanillins in the pipeline, TBJ 876, which is now sulfacolin, and TBJ 587. This is the next generation of dark vanillins, which were developed and designed to have improved safety and efficacy as compared to bedaquiline. They have similar structures, but have somewhat different number of physical chemical properties. They carry less potential for QT prolongation and more potent against MTB. So that, sorry if this is too small, they're about 10-fold more potent against both reference and clinical isolate strains in vitro potency assays. And importantly, they also display better potency against RV0678 mutants, which has very clear clinical relevance and implication. And this has been established also activity of both of the molecules against RV0678 mutant is comparable to activity of bedaquiline against the wild type TB. And this also bears out in the in vivo efficacy studies where they're more potent against RV0678, and that is the study published some time ago from Eric Nuremberger's lab. TBAJ 876, so surfactant, has been evaluated in the clinical trial NC009, which is a phase 2 clinical trial in drug-sensitive TB. The objective of the study is to evaluate efficacy, safety, and tolerability of 3 doses of surfactant in combination with bertomimir and linezolid. And there are critical comparator arms that included in the study, which is HRZ, standard of care, as well as bedaquiline, bertomimir, and linezolid. And the endpoint is the time to stable culture sputum conversion through week 8. And this has been presented at the Union conference before, and this generated a lot of interest. It's a very innovative clinical trial design, which is a dose-ranging study of surfactant, but in the context of the well-described TB and clinically used TB regimen that allowed us to avoid to bypass the conducting monotherapy dose-ranging study, and allowed for initial study of the efficacy in TB patients for 8 weeks, which is a shortened duration. It also will generate a couple of very good critical comparisons, which is a direct blinded comparison of surfactant with bedaquiline, as well as comparison with standard of care HRZ. So this is the trial design that, again, has been presented. The dose ranging surfactant was administered at 25, 50, and 100 milligrams in combination with bertomimir and linezolid, and in comparison with BPEL. And this is also notably a first study of bedaquiline, bertomimir, and linezolid in drug-sensitive patients. And again, this comparison to standard of care. And in the study, there was an option to stop at 15 weeks based on the 8-week status. So the results of NC009 now came through week 15. In this study, the dose response was demonstrated for surfactant with the potency and efficacy at 100 mg per kg bertomimir linezolid, and also the data is available for comparison of bedaquiline, bertomimir linezolid with HRZ. There were no dose-related safety issues in surfactant arms, with overall safety comparable to bedaquiline, bertomimir, and linezolid. The results. It's not moving. So the full results of the study will be presented as an oral presentation at the Union on Friday, and I very much hope that many of you will be able to attend that meeting and have a chance to discuss and see the outcomes. Okay.
So what's it, the arrow? Actually, he said, where is that? The oral
Yes,
presentation
the
will
oral
be
presentation.
Friday, November 21st from 9:15.
So also now I want to switch to TBAJ 587, our second direct quinoline, which has similar profile to TBAJ 876, but there are some differentiating features and the development of this molecule system continuing in parallel. TBJ587 has been evaluated through the platform, through the ERA for TB platform. And there we've completed phase 1 studies with ERA for TB consortium, both single and multiple ascending doses. There were no serious adverse events or safety concerns or withdrawals in those trials. And most AEs were very mild with safety profiles comparable to placebo. And based on the strength of these results, there are plans to conduct a phase 2 study within Unite for TB consortium in 2026. And conceptually, the design is the Part A and Part B study initially, which is Part A dose ranging study, followed by a combination of TBJ587 with novel compounds, which are part of the Unite for TB. And of course, the outcomes of the surfactant trial that we are analyzing now will inform the dose selections and planning for the study with TBJ587. And in addition, I will not talk about this, but there's a preliminary work going to evaluate also potential for long-acting injectable formulation for these molecules, and that's something that is very much ongoing right now. And so another clinical compound that's very interesting in our pipeline is telatabeg, or Q203. It was licensed, to be licensed telatabeg from Current in 2023 for the treatment against Mycobacterium tuberculosis. But we also recognize that it is active against both Mycobacterium tuberculosis, Mycobacterium leprae, and also Mycobacterium ulcerans. With respect to TB, we are working right now on the preclinical program to identify the companion drugs that could be combined successfully with Q203 to form a new regimen. And there are additional in vitro studies ongoing to evaluate activity of telatabeg against different strains with potentially different levels of beta-oxidase expression. But what we also recognize is that it has transformational potential for a treatment of both Buruli ulcer and Mycobacterium leprae and leprosy due to uniqueness of mechanism of action against this bacteria, which have lost all the alternative beta-oxidase pathways. So we have also, in addition, we're proceeding, there's multiple complementary pathways we're developing against TB, but also looking at potential against Buruli ulcer and leprosy. In terms of Buruli ulcer, we have completed the initial trial in Australia with 40 patients enrolled in 2024. And in this case, in this trial, telatabeg was highly potent, and there was cure observed after 4 week course, as opposed to 8-week course of standard of care. And following this, additional 8 patients were recruited. This treatment now is shortened to 2 weeks, which would be very short duration, which the community is very excited about. There will be there's plans to start a phase 3 trial in West Africa in 2026. And in addition, we are also considering telatabeg for leprosy. We are right now completing a series of preclinical studies to understand the optimal dosing and combination regimens, but the available data demonstrates that telatabeg is highly potent against Mycobacterium leprae. And we do have an 80-patient pilot study, clinical study, funded by DCTP, which will start in 2026. And the only thing that I do want to add, if I have a minute, is that in addition, you may see that in our discovery and development pipeline, we do have now inclusion of host-directed therapies in the exploratory way and pathway. This is something that we are pursuing right now, and we started with employing artificial intelligence to support immunotherapy and to identify pathways and targets that could be that have potential to combine with OTB regimens for shorter duration of treatment. And this is something that we're exploring now through several parallel approaches. One of them is working with the PEMIC Consortium, which is the consortium that is organized by TBVI, and we have a broad number of our compounds that came out from the artificial intelligence screen. And we also have additional integrated approach to target granuloma, which is now funded by DoD, and which we are pursuing with partners from both immunology and drug development fields. So it's very exciting. And what I want to mention is that we do have satellite sessions. So PEMIC has been progressing for several years right now. We have very interesting proof of concept studies that are completed. It is a multidisciplinary consortium which consists of PPPs, industry, and academia. And the goal of this consortium is to evaluate adjunctive immunotherapies and develop critical path for progression of HTTs. We have a satellite session at the Union Conference to highlight the progress and the achievements and the objectives of the consortium, and that will happen on Friday, November 21 and November 21st at 8 a.m.
Thank you.
I will stop.
We can take one quick question. Apologies, we're running a bit behind.
Thank you very much. Could you comment on the differentiation between 876 and 587? Is it the long-acting injectable potential, or maybe I missed it? And also, how long are you planning to develop both if they are so similar? Is there an inflection point where you plan, well, if 876 is good enough, then maybe we invest resources in another new mode of action or something like that?
Yeah. That's a very good question. So far, we have conducted sort of parallel evaluation, and that includes safety, in vitro and vivo safety and DDI assessments. And so far, both compounds meet criteria and are very progressible. They do have differences in physicochemical properties that may make them suitable for different indications. This is something that we're working on right now. So for instance, we could consider treatment of active TB versus latent TB, oral versus long-acting injectables. And that is where we may see the differentiation. But again, both compounds so far has been very progressed strongly, and we see it more at this point as a complementary sort of pathways.
Thank you, Natalia. We do have a number of TB Alliance people here tonight and this week, so if you have more questions, feel free to come find us. All right, we'll move on to GlaxoSmithKline. We have Alana Fernandez Alvaro. Thank you and welcome. It seems our clickers seem to have stopped working. Let me just check on it really quick. Okay. There you go.
Okay, great. Well, hi everyone. I'm Elena Fernandez. I lead the access planning activities within the TB drug pipeline, and it's a pleasure to be here representing GSK. Well, this is my conflict of interest in this paper form. And I will start by introducing briefly our unit, GSK Global Health. You have to mention that we are a team of 250 employees within the GSK mainstream organization. We do not seek to have return of investment for our programs in global health. Our success is measured by the positive impact in the health of the people living in low and middle income countries. So our objective is to impact the life of 1.3 billion people this decade, and which we hope we will achieve. And we are organized in 3 groups performing 3 activities. We do research and development of drugs and vaccines for infectious diseases in LMICs, but we also pursue equitable access of our GSK pipeline and portfolio, not only global health products, but our whole pipeline and portfolio as a company. And importantly, we also work on health system strengthening. We establish partnerships with key NGOs like Save the Children and Ref Health Africa, because we think that in order to achieve health equity, you not only need products, but you also need to create the infrastructure and know-how that enable effective use of those. So, and our efforts in the last decades have proven some successes. So In the left part of the slide, let me show you the products, the global health products that we've brought to market. I would just like to mention Mosquirix, because we are quite proud of the world's first malaria vaccine, combining the antigen RTFS and the GSK adjuvant AS01, which is being delivered to children in endemic countries, introduced in 11 countries so far, and the number is going to increase very soon. And more importantly, we recently partnered with Bharat Biotech, a manufacturer and commercialization partner for vaccines in India, with the aim to reduce the price of the product by more than 50%, with the aim to increase the access of the product, which is our default strategy as a global health unit. And in terms of pipeline, we have more than 25 vaccines and products. More than 1/3 of them are currently in clinical development. And now I'll take you through our TB drug pipeline briefly. So just to mention that we mainly pursue new modes of action. We want to deliver products that are active against isolates in the field that are resistant to standard TB drugs. We are focused on improving treatment adherence with shorter regimens, more tolerable, and also easier to co-administer with underlying treatments, like antiretrovirals, or easier to use in patients that have problems like addiction to substances. And we are also focused on simplifying the treatment journey, reducing the requirements for drug susceptibility testing, and ideally reducing the need for patient monitoring while on treatment, okay? And in the longer term, we believe that we have products that are strong candidates for the ultra-short regimen that combines orals and long-acting injectables. And in the next couple of minutes, I'll go through our two most advanced assets, Ganfevrol and Alpivectir, that have been previously briefly covered by Jody. So you've made a great presentation of the clinical trial design. So I'll provide an update from a slightly different point of view. Okay, so Ganfevrol is a first-in-class. It has a new mode of action. It's a protein synthesis inhibitor. It inhibits a new target, leucyl tRNA synthase. Our current data suggest no drug-drug interactions and very good tolerability. And Ganfevrol achieved clinical POC in a phase 2A study. You can see the data here at 1, 5, 15, and 30 milligrams compared with Rifampicin. So we have progressed the 20 milligrams one daily oral dose to phase 2 BC studies. In terms of phase 2 BC, you have to mention that it's being progressed within Paradigm, part of the Unite for TB consortium. We are currently testing 6 different combinations of Ganfevrol in 4-month regimens. And the interim analysis for all these studies will be available in the first half of next year. So exciting times ahead of us. And well, the results of this interim analysis, we will use them to plan for the phase 2C, which plans to use combinations 4 months or shorter, and also the phase 3. Okay, so Alpivectir is another first-in-class. Okay, it targets a new mode of action. It acts through a transcription regulator, and it increases the bioactivation of ethionamide. Probably all of you know ethionamide. It's a well-known TB drug, very fast, very cycle. But resistance to ethionamide leads to the need to increase the dose, and high doses of ethionamide are not tolerable. So what we see is that the combination of Alpivectir and ethionamide allows the dose reduction of ethionamide. And also, we are able to see activity in strains resistant to isoniazid and ethionamide. So we are able to reduce the dose and overcome resistance. So we've seen all this mainly in preclinical studies in collaboration with BioVersys, a Swiss biotech. I think the pointer is not working, but you can see in the in vitro studies, in the killing curves, you can see that Alpivectir combined with ethionamide in the blue line achieves the killing effect of isoniazid, and it allows dose reduction of ethionamide of up to 10 times. Then if you move to the animal studies, you can see a similar effect. If you compare the black bars of isoniazid with the gray bars of ethionamide and then the blue bars of the combination, we see a potential more than 3-fold dose reduction of ethionamide, and again, similar activity to isoniazid or better. And now what happens in patients? So we have tested different doses of alpivectin and etironamide in 2 phase 2A studies, which have been completed and read out, and results will be made available very soon. So stay tuned because, well, we will soon see how these preclinical effects translate into humans. So this is my last slide, just to mention that we continue filling our pipeline. We have TSK-30, another compound acting through a new mode of action that was candidate selected this month. And we have several other assets in earlier stages that we hopefully will continue progressing. And thank you very much to all our collaborators and co-founders, and I'm happy to take questions if any. Well, this should be a big thank you. It's a big TSK, but thank you.
Any questions for Alana? If not, thank you so much. A lot of great progress. So our next and last speaker is someone who will look very familiar to you. So yeah, I know it is working out. There you go. So representing Smart for TB, Eric Nuremberger.
Thank you again. So I'm going to give an update on the Smart for TB consortium and the 3 clinical trials that are ongoing here. As you may be aware, we were originally, this was a consortium sponsored by USAID that has subsequently been taken up for sponsorship into the US State Department, where timeline is now running through July of 2027. We're now focused on two specific thematic areas, diagnostics and therapeutics. Within the therapeutics area, we have 3 clinical trials that are either ongoing or preparing to get started soon. The first I'll talk about is SMILE TB. This is being led by Nicole Salazar-Austin at Hopkins, along with collaborators, site PIs from 7 different countries that you can see pictured across the bottom here. This is investigating a shortened regimen for drug-susceptible TB in kids. Here is the design. So children under 10 will be randomized to receive a control regimen, which is essentially patterned on the SHINE regimen, where children with less extensive pulmonary or lymph node disease will receive 4 months of standard care, and those with more extensive disease will receive 6 months. Where the intervention will be the isoniazid ribapentine pyrazinamide moxifloxacin containing regimen for 8 weeks with no stratification. Follow-up will be out to 72 weeks. This trial actually started enrolling and enrolled a few participants early in January before the stop work order came from USAID, and then picked up again in May, and now has enrolled over 40 children. There will be an initial interim analysis of rifapentine and moxifloxacin PK, seeking to achieve targets established for adults in study 31, and then move on to stage 2, hopefully with the same doses, and enroll up to 850 or so participants. The second trial I'll talk about is PRISM TB. This is for adolescents and adults with rifampin-resistant TB. It's a stratified medicine trial that's being led by Gustavo Velasquez, Patrick Phillips, and Rada Savic at UCSF. It's being run and will be run in 3 countries, Peru, Uganda, and Mongolia. This has been redesigned as a 2-stage trial. Stage 1 is funded by the US State Department. In this study, these will be individuals with fluoroquinolone susceptible RIF-resistant TB. They'll be enrolled randomized 1-to-1-to-1 into a standard of care control arm, or one of 2 investigational strategies. The investigational strategy, both of these will look at BPALM and different durations of the BPALM regimen, and all participants enrolled into those investigational arms will be stratified by a risk score that is based on HIV status, cavitation on chest X-ray, expert cycle threshold, as well as age, sex, and BMI at enrollment. In investigational strategy 1, those in the high risk score category and low risk category will both get 17 weeks of treatment. Investigational strategy 2 is actually stratified in stage 1. The lower risk score participants will receive 13 weeks versus 24 weeks for the higher risk scores. And then those data will be analyzed and will be used to support stage 2. This stage is currently still seeking funding to conduct. And here the BPALM investigational strategy will be devised based on the preferred durations for individuals with the high risk or the low risk in stage 1. So there are several different combinations of durations for individuals based on their risk that will be informed by stage 1. The primary outcome in stage 1 that will be used to help establish the regimens for stage 2 won't be the sole outcome, but will contribute to that, is a desirability of outcome ranking score. And you can see that that takes into account and essentially ranks outcomes based on 3 parameters, efficacy, safety, and tolerability. And so just interpreting this, if you go across the top, you can see that the best outcome, the ranking of 1 would come from cured or treatment completed, no grade 3 AEs, and no treatment changes required. And then every deviation from that would lower the DOR ranking. Here's the timeline, and stage 1 has just started in the last month or month and a half with enrollment in Mongolia. The first few participants have been enrolled. They will be followed up for up to 28 weeks during stage 1, and then extended follow-up thereafter, hopefully with stage 2. You can see the timelines here. Finally, I'll talk about the BREACH trial. This is a prevention trial looking at a short course bedaquiline regimen. This being led by Sonia Krishna and myself at Johns Hopkins and colleagues in Peru, Uganda, and Tanzania. This has also been, the design has been revised into a seamless 2-stage study. This will be open label, a non-inferiority study, enrolling individuals who are contacts of either drug susceptible or RIF-resistant tuberculosis who fall into a high risk category in terms of their probability or risk of developing TB disease, as well as persons living with HIV in relatively high transmission settings. Those who are presumed to have RIF-resistant TB will be randomized to a 4-week bedaquiline regimen or the control 26-week levofloxacin regimen. Those who are presumed to have drug-susceptible TB infection would be randomized to the same bedaquiline regimen or the 3-HP regimen. So the goal of enrolling these populations is to seek a short course pan-TPT regimen, if you will, that could be applied irrespective of exposure to RIF-resistant or DSTB. So here's the schema for stage 1. This is based largely on safety and tolerability based on evaluating safety and tolerability of the 4-week bedaquiline regimen, the primary outcomes being permanent discontinuation due to treatment-related AE and on-time treatment completion. Those primary outcomes will be assessed at different time points, 18 weeks or 36 weeks, depending on the arm, with 150 participants per arm. And really there, the primary outcome is really looking at just estimating the safety and tolerability of the bedaquiline regimen relative to these others. After interim analysis, and this will be seamless, participants will be enrolled into stage 2, which is the original efficacy and safety trial. Here looking at incident TB as the primary efficacy outcome, with follow-up to 72 weeks and a sample size just over 2,500 participants. So this is just a little bit of an update on the status of each of the trials. I mentioned SMILE has been enrolling in Uganda, although interrupted for about a year, with over 40 participants recruited now, and is opening in other sites here soon. I believe Indonesia and Zambia have already opened. PRISM is open in Mongolia, soon to open in Peru, and then Uganda. And BREACH hopes to open in Tanzania before the end of the year, and soon thereafter in Peru and Uganda. So with that, thank you.
We have time for one quick question.
Thank you very much. Very quick. So since you want to test 1 month bedaquiline, why did you choose the 3 HP as a control and not the 1 HP?
So that's a good question. We originally actually were including both 3 HP and 1 HP as options in the trial, but in an effort to streamline the trial, given reductions in funding and timeline, we constrained ourselves to 3 HP, which is better established and
because of safety problems, because of
not intentionally because of safety problems or tolerability problems, but we are mindful that that has been associated with some tolerability.
At least you would have avoided
I mean, it was going to be an open label trial from the beginning, but we had sites that were already up and running and using 3 HP, but not necessarily 1 HP, and so it was really just for simplicity.
Thank you so much, Eric, for 2 presentations. So I just want to take a minute to thank all of our speakers. Let's see. Thank you all so much for being here in person. This honestly could never have happened without the support from the TV community. And as I'm closing the working group on new drugs meeting, I also want to invite up the vaccines and diagnostics group. Together we put together this symposium today, and for those of you who've been here since the morning, wow, really just thank you for participating. It's been such a great session. So yeah, here, as we close the WG&D, we can also close the symposium. Morton, would you like to say a few words?
Hey, thanks. I have a list of people I'd like to say thanks to. Of course, the organizing team, Adam, Daniela, Said, Jennifer, Dave, Wayne, who has been putting some strings behind in his spare time because there's start to be partnership issues. Technical team at Find, who has been live streaming and been on the whole day. Yavi, TB Alliance, the Nivi team, Henriette, Mette, Nina, and yeah, all of you for hanging in there.
Thank all the audience who have been in the room, some familiar faces for the whole day, and anybody who's still on the Zoom call or can't see, but thank you for your patience through that, and an exciting year. I look forward to a lot to come in the years ahead as well.
Just I'll reiterate thanks from the Vaccines Working Group. We appreciate everyone being here and your interest, and look forward to bringing you more updates and continuing to work with you.
Nothing to add. Just thank you for staying until 7 o'clock tonight, and thank you, Morten, for hosting us. That was and exactly, Mary, and it was a great job. Yes, thank you.
It's been fun. It's been fun. So we have no budget for drinks, but we invite everybody who has time and interest in a snack and a beer or a glass of wine to go to this location, use the QR code there. I hope they are ready for us.
Thank you, everyone.
Already there.
Thank you.